All right, welcome back, everybody. So today I thought we could go back to our phenotyping and we can use InSituType again and apply a cell profile, what they call it, to our data and derive some of our phenotypes. I will show you the link to the cell profile page, and then I'll also show you kind of the tutorial that we're working on, which is on the InSituType GitHub repo. So let's just jump right in. If we head on over to the cell profile GitHub repo, we can see that we have a lot of instruction here about how they had done some stuff. But what we want is we actually just want to get the brain atlas. So there's the Allen brain atlas, this guy here. And that's the one that we're going to apply to our data, or take our data and apply it to this and derive the phenotypes that our data has and know what types of cells are in our data. And in order to do that, we have to click on it and get the raw. And then if we just right click and then copy the link, what we're able to do is use this in our code and then surround it with URL. And we're actually able to just load the data right in with R. We don't have to download it to our machine or anything. Just load it right into R. And then the example that I'm going to be showing today is the non-small cell lung cancer and then the vignette. So if you want to download that, kind of look through the different steps and stuff there. Basically going to be doing the exact same thing except applying it to the brain samples that we have, the human frontal cortex. And yeah, so let's get started. The three libraries that we're going to use today is InSituType, which we had initially downloaded way back at the very beginning for phenotyping, and then Seurat in order to read in our Seurat object, and then Tidyverse. So Tidyverse is going to allow us to do some of that piping again and just make it a little bit cleaner. Next, we're going to load in the SC transform data that we had done back a while ago, and then we're going to load in our human brain atlas from the cell profile page. So if you have different cell or different tissue, you can go ahead and use anything else from here and should be good to go. And what we're going to do is actually considered supervised clustering. It's not semi-supervised. We're going to get just the phenotypes that are in the reference, whereas if you do semi-supervised, you'll get those that are in your reference plus like A, B, C for unknown clusters. This is going to be supervised. Once we have that, inside of this load here, it's going to load 4 different things. It's going to have our cell groups _ class, metadata, and then the profile matrix, which is actually a data frame, not a matrix. And if you look at the metadata, it'll show you kind of where the data came from. Whoops. And then you can kind of follow that down if that's of interest to you. From there, what InSituType is going to need, similar to the first time, is going to need our expression matrix, and then it's going to need the false positive, kind of like the background noise of the different cells or the different probes in the cells. So in order to get the actual counts, so this is the actual counts, not the transform data, it's going to be an object, assays, RNA, and then counts. So if we go ahead and look at this, it's actually integer values, 0, 3, 2, so on and so forth. And we need it to be in the matrix format. And then we're going to rotate it. That way we have cells in our rows and genes in our columns. And then for the negative probes, what we're going to do is we're just going to take and find the average count for the negative probes in our sample. So if we go ahead and take a look at this, each cell will have a number. So that's the average number of negative probes identified across the -1, 2, 3, 4, so on and so forth for each particular cell. Next, a really nice thing about this particular type of run is that you can do cohorting, or basically you create clusters with some observational data that you have, so you can do staining. Here is using a couple of different columns within our data. If we go ahead and look at what columns these are, histone, G, rRNA, GFAP, and then DAPI. So using these values, we're going to go ahead and create some clusters of our different cells. If you have a particular immunofluorescence marker in here, you can go ahead and kind of manually threshold that. So you can say these cells are high for like pancytokeratin, so they're going to be tumor cells. These cells are low for pancytokeratin, so they're stromal cells. You can create different combinations and stuff that way. If we go ahead and look at this, there's an awful lot of clusters, so it's hard to say if this is going to be informative to the InSituType clustering method, but it's better to have it than not have it, I would say. If anything, it just provides zero weight to the cohort that it calls. Otherwise, it can be informative, saying these two have similar gene expression profiles, so they're going to be the same cell type. Yeah. Then we can actually feed this into the InSituType ML formula, or function. So we're going to feed in our count data, and that's where our cells are in our rows and the genes are in columns. We're going to feed in the negative mean, so that's the average number of negative probes identified within each cell. Then we're going to feed in our cohorts. That's going to be created, like in this case, with whatever those markers are, or it's going to be something that you provide. And then we're going to feed in our actual profiles. So let's look at our profile matrix that they call. So in our columns, we have our cell types, and then our rows, we have our genes. And what this is it's each column is its own cell type, whereas if you download a like single cell atlas, you'll maybe have 1,000 cells that are astrocytes. What this here is it's the average expression for a gene in that cell type. So we only have the same number of columns as we do different cell types. Then once that's run, we get this supervised clusters list object. So we have clusters, and this is going to be just a character. So each cell has, let's do head supervised clusters $clust. So each cell now has an assignment. We have the posterior probability assigned to that assignment, and then profiles. So that's just the gene expression intersection between our cells and the profile. And then negative log likelihood, so on and so forth. What we can do then is we want to store back in these assignments in our Surrat object. That way later on we can go ahead and plot. So an easy way to do that is to do object and then $, whatever you want to call your new column. Here I called it InSituType IDs, and then we're going to assign it the cluster IDs that are down here at the bottom. So we're going to have a new column in our metadata that's called InSituType IDs. So if we do call names at metadata, then we can see we have our InSituType IDs here, which are going to be our new phenotypes. Something that I did notice, and if we scroll back up here, we'll probably see that warning. There's a lot of genes that are not present in the actual profile that are in our data. And one reason for that is going to be that there's custom probes here. So there's a couple hundred custom probes that the name's custom, so it's not going to intersect with any known gene ID. But then there are also some that have different names in the in the Cosmics data than in the reference data. And I'm not 100% sure why this is, but I have a couple examples here to show what I mean by different names or different IDs. So ITGA5 is what's in the expression data. So that's what's in our Cosmics data. But then in the reference and other places that I've seen, so like the single cell reference that I built with the kidney cell atlas has ITGAV as the gene symbol instead of ITGA5. So if you have a really high number like this, where they don't have the same ID, so we have so many missing names in our data that are not in the reference, I would go back through and make sure you can change the ID in either the reference, or you can change it in your data and then put it back in. I would just change it in the reference. That would make it a little bit easier. Another one is ITGA10. So the integrin alpha 10, and In the reference data, and I think this is the more correct one, it's ITGAX, so Roman numeral 10. And if this is something that comes up in your data, I would talk to NanoString or whoever the vendor is that provides the data if you're using Xenium like 10x. But that's something to keep in mind too. You might have really low, I shouldn't say low, depending on how many genes you have in total. So here maybe, I don't know, 20% of them didn't intersect, but we have a high number that did. Just something to keep in mind. And yeah, what we can do is with our data, so that's going to be this supervised clusters, remember, we can collapse all the cells down into the profiles. So we have 19 different cells of our reference, and we have all of the cells in our data. If we collapse our cells down into those 19 different classes, we can plot the expression of different genes in the data there. So if we go ahead and look at this, make it a little bit bigger so we can read the phenotypes there on the bottom. We can see that we have a really hot block here on the left-hand side, which seems to be the different layers minus L5 here and L6B. For somebody who, well, maybe L6 doesn't belong in there. For somebody who is more familiar with the layers of the brain, this might make a lot more sense than it does to me. But this is another way to visualize if you know the different phenotypes in your sample. So you might expect the immune cells to cluster together in this sort of thing. You might expect particular tissue cells to cluster together if you know that you have very distinct lung cells. So this is another QC look that you can just visualize and see, okay, yes, my cells are making sense for the gene expression that was assigned to them. Next we can plot our cells, the expression over the UMAP that we had created with the SC transform data. And I did that with the UMAP. And we can see here that we have a bright blue, might be microglia or oligodendrocytes up here. We have some either astrocytes or the VLMC down here. Green is in the middle, different layers, and then yellow over here. So we can see that the clustering did a pretty good job. So remember, this is only a 2D representation of a high dimensionality or of high dimensional data. So some of them might kind of overlap in the 2D space, but so we can visualize that. Then we can also visualize a flight path plot. And what a flight path plot is, at the center of these clusters, you have a posterior probability. So the higher the posterior probability, the more confidence that you have that the assignment is correct for that particular cell. And we can visualize this. And those that are falling between two centroids have a lower probability or a lower confidence that they belong to any particular group. And we'll see kind of the trail between the two. Oh, that's a really bad looking plot. Let's try again. Yay for random seeds. It's not providing the IDs now. Let's see if we get back. There you go. There we go. So you can see the posterior probabilities for our different groups, and those that are falling kind of in these trails, if you will, have lower confidence as to what cell type they actually belong to. So somebody who works with brain might understand these and say, okay, there's some transition between L5 and, I don't know what this is, whatever this one is, or SST and whatever this one is sort of related. So the idea that there are cells with confidence maybe in between makes some sense. But then we also have like this parasite out here that is very isolated. We have the L5 that's very isolated. Astrocytes are very isolated. So this just gives you another way of visualizing the confidence that your different cell types have for the IDs that we are assigning to them. And then we can plot just like we did when we were exploring our data. Let's see if I can shrink this down a little bit, get those points a little bit closer together with our new phenotypes. So we can see that there's this hotter region like we had originally seen of the blue cells up in this upper left-hand corner, and then it's more vacant of those blue cells here in the bottom right. And since this is a study where all the fields of view came from the same slide, if we were to kind of spend some time and plot all the fields of view next to each other, we would expect that this structure that we're identifying is following through into other fields of view wherever they happen to be. I think this one might be in the upper left corner, so this structure might be on its own, but it is something that could be plotted all together and create one big tissue image or plot to visualize all of these different cells together. But that is supervised clustering using InSituType. I will put a link down into the description because I think this is a very powerful resource to have all these cell profiles available. And yeah, it just eliminates the need for every person to go and create their own. Plus, it's harmonized now. So if everybody uses the same profile, it makes it easier to compare across different studies. So yeah, let me know what you guys think. Bye.